Well, hello. My name is Stan Trauth, and I am presenting on the morphology of Rathke's glands in the alligator snapping turtle. This research is a part of my ongoing study of these glands, which are often called musk glands or scent glands in turtles. I have published 4 papers on these glands in the last 13 years, and 3 of these studies have been published in the Arkansas Journal of the Arkansas Academy of Science. Well, to begin with, it was Rathke who in 1848 identified these glands in the terrestrial turtle and a terrestrial turtle in Europe. And so he is honored by having these glands named after him about 100 years or so later. Here are several facts about Rathke's glands. They're found widely among recent turtles and are thought to be homologous among all turtles. They are fully formed and functional in hatchlings. They're specialized for the production and episodic forceful extrusion of a smelly substance, often smelly substance or secretion. 1 of 5 pairs of glands will occur, and there are 9 different patterns of external pore numbers and locations among turtles themselves. The presence of Rathke's glands is thought to be a basal condition for all turtles. Their presence, primarily in terrestrial turtles, is presumably derived from the ancient or fossil turtles. In fact, you can see a pore in the shell of fossil turtles where the gland external pore is found. So gland morphology in most turtles, including chelydrids, except for the snapping turtle, which I studied in 2012, is poorly known. So we know a lot about some turtles, but much of the details of many turtles is still unknown. So here is the where these glands occur. In A is the common snapping turtle, and in B is a soft-shell turtle. And if you look at A, and there are 4 glands on each side, the axillary gland, and then there's 1, 2, and 3 inframarginals. Here is in the first figure here shows a snapper dissected, showing the axillary gland and the first, second, and third inframarginals. Gland length is about 15 to 20 millimeters. Gland width about 8 millimeters, and gland height is about 4 to 5 millimeters. Now, here's the first in a series of micrographs of the glands, and if you'll note, most of the gland is comprised of striated muscle. So all of this area here is striated muscle. Here's the gland proper and the lumen of the gland. And if we move to B and C, you can see here is the lumen still, and there's the epithelium that is producing these secretions. In C, you can see it's a singular layer, not well developed in the alligator snapping turtle. If we move to D, which is a plastic section, as opposed to A, B, and C being paraffin sections, we can see one of the 2 types of secretory vacuoles that are produced by these cells lining the gland proper. This is a blood vessel. Here is the striated muscle down here. So I was somewhat surprised not to find large numbers of these secretory vacuole type 1s. As you can see in this next slide, here is the common, we'll call it the common snapping turtle with these type 1 secretory vesicles, very prominent everywhere. And type 2 secretory vesicles, which are not identified here, are these other structures I'm pointing to right now. And you can see them over here in A, which is showing the type 1 secretory vacuole, well-defined, a glycoprotein, and then the secretory vesicle type 2, and these are often evacuated spaces because of the lipid components which are washed out of these particular vacuoles during histological processing. In B, you can see within this the holocrine cell itself here, the type 2 secretory vacuole. Here's a type 1 secretory vacuole right there. Here it is down here in this particular cell. So these cells are released, are produced and then released from the epithelium. And then they, as they move away, they begin to break down and release their products into the interior of the lumen. Now, these are not from the alligator snapping turtle. These are from the common snapping turtle. As it turns out, as I began to study the alligator snapping turtle, and I did use 3 specimens in order to try to find large numbers of secretory vacuoles, they just were not present. In A, you can see a single one there, and there's one there and there, but not very many present. In B, which is a magnification of A, you can also see this secretory vesicles type 2s, a line or series of them right there. So there are no more than just one per cell, whereas in this secretory vesicle type 1, you usually just have a single large vesicle or vacuole, excuse me. Here are the holocrine cells that produce these secretions, and here again they don't show the large amount of cytoplasmic material like the cells in the common snapper, for example. In C, you can see a holocrine cell being released, that's been released, and you can also find secretory vacuole type 1 and secretory vacuole type 2, and the type 2 here is releasing its material into the lumen, very evident there. And then in D, what remains after these cells begin to die and elaborate their materials, the nucleus is going to remain. You can see several nuclei there. The best way to study these particular holocrine cells and these glandular products is by electron microscopy, but I was not able to do that during this particular study. Still see the structures, still see the secretions, but not in the detail you can with electron microscopy. That is the transmission electron microscopy. These are scanning electron micrographs, which simply show the massive amount of striated muscle associated with these glands. A lumen is present of the gland proper, and you can see the lumen there, even though it's not a continued lining here. And in each case, you can see the lumen, but not much of a gland proper itself. Over here, you're only seeing one surface of the lumen, and then in this particular view in D, you can see more or less the entire lumen of these particular glands. Why these are not holding together better, I have no idea. Why would I not see the entire lumen? It still remains to be understood. Now, if we move on to the epithelial linings, there are some interesting aspects of the linings. For example, these intercellular bridges, little tiny spokes you can see connecting all of the cells, very prominent in A, and they are prominent in B also. We move to C, we can find a few of the secretory vacuole type 1s that are present, but not many. And I'm not understanding why there aren't many to be seen. And this was the case with the 3 specimens that I examined, and I was not going to sacrifice a 4th specimen in order to try to find these. They're just not numerous. Over here in D, you can see how the cells, holocron cells, have attenuated themselves, which is sort of an interesting feature of this particular gland. And then there are these secretory blebs, which are released by the cells. They're quite numerous, and that's a product of these holocron cells. So this gives you some idea of the detail of these glands, but still nothing quite as dramatic as what you would find in a common snapper or in the other studies I've done on other turtles. So it's a interesting animal to study, but I was not totally happy with the results. That is, they were not as conclusive as in other studies. So that concludes my study.